Okay, great. All right. Yeah. So good evening, everyone. um, Great to see you all. So my name is Sean Edington, and I'm a postdoctoral associate in the chemistry department at Yale. And I'm going to share a little bit with you tonight about some of my, my former research in the field known as physical chemistry and biophysics. But first, I want to kind of just start off with a little bit about who I am and where I am in my career. I've been kind of all over the country during my academic career, but it turns out I grew up right here in Connecticut, in Storrs, right by UConn. So has anyone been to Storrs? Does anyone know about Storrs? It's a beautiful town. The funny thing about Storrs is that it's pretty much the University of Connecticut and then nothing else. But I kind of like it that way. It's kind of quiet, it's relaxing. So I went to high school in Storrs. And then I moved to Virginia, to the University of Virginia for college. And there's, it's got a beautiful campus. And that's when I got my first taste of physical chemistry as an undergraduate there. And I'm showing you in the bottom right corner, this is one of the instruments I worked on when I was an undergraduate. And we have to go into too much about what it did, but it's called a thermal desorption spectrometer. And a lot of people ask, what's up with the tinfoil on your machine? And the reason for the tinfoil is that in order for these instruments to work very well, the inside has to be super clean. It has to be empty like outer space. And so it turns out that molecules hanging on the inside of the machine will actually interfere with our experiments, even if it's already under vacuum. So to get rid of those molecules, we have to heat them up to drive everything off. And that tinfoil traps near the surface to help it get really hot. So then for graduate school, so I got my college degree, then I went to Princeton University down in New Jersey for my doctorate in physical chemistry. And I'm showing you up on the left-hand side there. It's another kind of familiar looking machine. That's actually a thermal desorption spectrometer as well. But this is one that I didn't just use that one. I built that one myself. And I use it to do some experiments. Even so, I think it's important to mention, even if you go into science, science doesn't just all work all the time. And while I was a graduate student, I played a lot of soccer with other graduate students down at Princeton. And so that photo I'm showing you down there, every single member of that photo of my soccer team was also a graduate student at Princeton. So, we got out and we got some exercise as well. And then finally, my last step before moving to Yale is if you want to become a professor, it's not actually enough to just have a PhD. You also have to do additional training after you get your doctorate. And so I did some of that down in Texas at the University of Texas at Austin. Beautiful place to work. I played soccer there as well, though I don't have a picture of that here. And during that time, I built this device in the bottom right-hand corner. It's called an ultra-fast spectrometer. I'm going to be telling you a little bit more about that during the talk. And so this is the device that we use to study proteins and how they work in our bodies. Okay. So I mentioned earlier that I'm a physical chemist. And, if you're not too familiar with chemistry, you might wonder, well, what does that mean? Physical chemist, organic chemist, biochemist. And so there's kind of a sliding scale of chemistry. And from my perspective, at least, and I see someone ask, what do physical chemists do? And great question, because I'm going right there. Kinds of chemistry that you might be more familiar with from TV shows usually involve glassware and like a stir plate and fluids going around in tubes. And I think often those kinds of chemistry fall into the province of organic chemistry or biological chemistry. And so in those cases, you're using this kind of familiar chemical equipment to look at large, complicated molecules. And I said there is a sliding scale. And so the sliding scale is you kind of look at the larger molecules or synthesize these larger molecules with, and I see someone's mentioning Isaac Asimov, one of my favorite authors. So that's cool. Sliding scale, the bigger your molecules are, you kind of use these more typical apparatus. But I'm at the other end of the scale, where we look at really small molecules with really crazy looking instruments and machines, which is another word for apparatus, that word I was just using there. And so I'm showing you on the right here, this is yet another machine that I built during my career. And this one is a machine used for studying the kinds of reactions that happen in your car's catalytic converter. And so it turns out that, every car has that catalytic converter. So we care a lot about that chemistry and what's going on in there. And so I just want to ask in the chat, while I'm going through, does anyone know what a catalytic converter is? What does that do? Getting a nope, no idea. No, all right. So I think that's good because I have an opportunity to actually provide some useful information here. So what's a catalytic converter? So if you look underneath your car, and if you're to do this, make sure the engine is off and the car is securely parked with the emergency brake engaged. But if you look underneath your car, you'll see the exhaust pipes and everything going underneath it. And you'll probably also see a rectangular block that looks something like that. And that is a catalytic converter. And what a catalytic converter does is, when your car is running, it produces a lot of poisonous gases. And that's why you never want to run your car in an enclosed garage. And part of what a catalytic converter does is it converts some of those really toxic gases to gases that still aren't great, but aren't quite as toxic. So for example, your car's engine produces a lot of carbon monoxide, which is very poisonous, and is very dangerous to people, even in pretty small concentrations. And a catalytic converter converts that carbon monoxide to a different molecule called carbon dioxide. And of course, carbon dioxide is still not great, and it's responsible for global climate change. But it's not as poisonous as carbon monoxide. So we want that reaction to happen before the exhaust gets out of the car's tailpipe. And so if you look down a catalytic converter, you can see that it's got all these tiny little channels. You guys see that? Look carefully, you can see there's lots of little holes. And those little holes cause the gas, it kind of forces them, the gas molecules, to interact a lot with the surface. And it's called a catalytic converter because inside all those little holes, there's a platinum surface. And platinum does something this is where the term catalytic converter comes from. It carries out catalysis, which means platinum makes a reaction go, but it doesn't get consumed itself. So a very small amount of platinum can convert a lot of carbon monoxide and other gases to carbon dioxide. And so if you cut away at the catalytic converter, you can see that honeycomb structure exists throughout the whole length of the catalytic converter. And so the whole point of this thing is to maximize the opportunity that carbon monoxide and other poisonous gases have to interact with that platinum surface and get converted. All right. So that's a catalytic converter. Now I've shown you, right, so far already, I've shown you many things that I got to build, as a scientist. And I think that's one of the greatest things about physical chemistry. That if you go into physical chemistry or a lot of other related fields like physics, you get to build all kinds of things. And so I already showed you this device that I built as a graduate student. And so this is what's called a laser spectrometer that uses essentially the color of light to look at what molecules are doing. In this case, in the gas phase. This is also, I showed you already, this is another device I built as a graduate student called a thermal desorption spectrometer. I don't want to get too much into that because it's not really related to the body or biomolecules. Another thing I built, this is a little sample holder, and this sample holder actually goes down inside this chamber in the middle. And what we use the sample holder to do is we can use it to heat molecules up to hundreds of degrees, and we can also cool it down very cold. I think in one of your earlier talks, someone had mentioned using liquid nitrogen to cool things, and that's exactly what this sample holder does. You can see there's this aluminum tube that goes into a copper block here, and we actually pour liquid nitrogen down into that aluminum tube, and it cools down the copper block, and it can get whatever is sitting on that sample holder to a very low temperature. And in case you want a factoid to take away from this talk, the boiling point of liquid nitrogen is 77 Kelvin, which is a unit of temperature that scientists use a lot. And so that's how cold, approximately how cold we can get things with liquid nitrogen. Here's another thing. I can't take credit for building this. This is actually an electron gun. And electron guns are used to hit surfaces with electrons. It's a way you can analyze things in a physical chemistry lab. And I didn't build that, but I did get to repair it. It's a very complicated device. You can see in the back there's some schematics with its electronics and physical dimensions there. And then finally, I mentioned earlier, some of these experiments I did in graduate school were in the gas phase. But for my first postdoc at UT Austin, we're looking at biomolecules in the solution phase, which is an important phase because that's the phase these molecules actually are in when they exist in our bodies, right? It's not just gas all in your bloodstream, it's liquid. And so this is a device we can use to study biomolecules in the actual liquid state, which is very important. All right, so we're talking about studying biomolecules in their natural states. And of course, so we have to discuss a little bit about cell structure. And I'm not a biologist, so I'm not going to go too much into the structure of the cell. But of course, the structure of the cell is very complicated, and there are all kinds of components. Very importantly, you have the cell membrane, which encircles the cell and serves as a border between the cell and its surroundings. And then, of course, inside the cell, you have all kinds of little organelles, or kind of like little factories that float around the cell and carry out, various functions. And so you may have heard of the mitochondria, or the lysosome, or the nucleus, various structures in the cells. And so when we're talking about cells, right, we can talk about those structures, the organelles and everything, but those are still, remember, I mentioned as a physical chemist, I deal with small stuff. I try to deal with small stuff because I can't handle big stuff. That's a problem with physical chemistry. You can't deal with big stuff. And so all of these organelles and cellular machinery, these can get very big. So what I study, or what I studied down in Texas, are proteins. And so now we can go to breakout room for maybe 2 minutes. And I'd like us all to talk about, when we're talking about cells and someone says proteins, right? What do we mean when we're talking about proteins, and what do these proteins do? So let's head to breakout. So anyway, I hope you guys all had a good breakout, and I hope you had some good questions. And so the short answer to that kind of discussion is that, proteins do a lot of things in our cells, but I would say the short answer is that proteins are little molecular machines that our cells create and then employ. And proteins do all kinds of jobs in the cell. They produce energy for the cell. They take care of waste for the cell. They allow molecules to move in and out of the cell. And to answer, I think another question that was in the breakout is, where are these proteins located? And the answer is they're located all over the place in the cell. But different proteins of different types gather in different parts of the cell. So for example, proteins that are involved in producing energy for the cell are concentrated in the mitochondria, which, there's a meme, the mitochondria is the powerhouse of the cell. And that's very true. And so proteins whose job is to make energy hang out in the mitochondria. Whereas, on the other hand, there's some proteins that get rid of waste, that chew up waste molecules that they don't poison the cell. And for example, these proteins hang out in an organelle called the lysosome. But the proteins we're going to focus on are proteins that actually are concentrated in the cell membrane. And so you can see I've highlighted the cell membrane in a red box there. And so, right, proteins are molecular machines that drive all biological processes. And let's zoom in now on that cell membrane. Right, so here's a cutaway view of our cell membrane. If you snipped out a rectangular or a square portion of the membrane, this is what it would look like. There are many different molecules that make up that cell membrane. And I want to focus on this thing here, this little channel looking thing is an embedded protein in the cell membrane. And so embedded proteins allow things to flow into and out of the cell. And so that's very important because your cells, they need to consume certain molecules, and they need to get rid of other molecules. So these embedded proteins are super important for that task. And so these embedded proteins, though, they don't just kind of do the same thing all the time. Just like if it's cold out, you would sense that it's cold out and you put on a jacket. The cell can sense its environment, or I should say the proteins can sense their environments, and they change how they behave based on the environment. And I'm not going to talk too much about diffusion. I mean, that's a whole nother, that would be a whole other, half hour, a month. You can talk about that forever. But yes, diffusion is very important. And that, proteins, it turns out the cell membrane is not fixed. All these proteins and other components of the cell membrane are constantly moving around inside the cell membrane. It's a very dynamic medium. but so anyway, all of these proteins are constantly sensing what's going outside of them. And based on what they sense, they change their behavior. And so that's exactly the kind of process that I study in my research. And so let's look, we're zooming in now on that channel protein. And if we look really careful, we can see that channel protein bridges across the cell membrane. And I'm showing it now in what's called the closed conformation. And conformation is a technical term that physical chemists and biologists and scientists use to describe one particular layout of a protein. And so it's in the closed conformation right now, nothing can get through. So maybe that channel controls sodium going into the cell, and the cell's got enough sodium, so they don't need any more sodium, close the gates. But now a molecule that tells the protein, hey, we need more sodium in the cell, open up the gates, right? So when that molecule comes along, the cell opens up, and now it allows sodium to come in. So that's part of how a cell or how a protein senses its environment and changes its behavior in the right way. Okay. And so I just want to mention, so these are generally called cell member, or I should say, these are generically called transporter proteins or ion channels, and they change their shape to allow molecules to get into or to escape the cell. And yes, someone asked, can you compare the epidermis of the human skin to the cell membrane? I would say yes, you could. I would say, however, the epidermis is a little more focused on keeping stuff out than letting stuff in, right? But yeah, when your body gets hot, your epidermis opens up to let some sweat out so it can cool you off. So there's a very good analogy there. All right, so we're talking about cells that transport things. We're talking about how cells sense things. And calmodulin is very specifically the protein I study, and that's called a signaling protein. Calmodulin doesn't actually let things into or out of the cell itself, but what it does is it senses changes in how much calcium is around. And based on those changes in calcium, it will tell other proteins what to do. And so in this film strip, I'm showing you on the left is kind of calmodulin's closed conformation, you might call it. And then at the end, what happens when it senses calcium, and calcium attaches onto calmodulin, excuse me, and then it changes its shape. And you can't see the calcium atoms in the calmodulin right here in this film strip, but we'll see another view of calmodulin soon that will show us that. Now, I've shown some question marks in the middle of this film strip. And the reason for that is calmodulin works super duper fast. And so one example of how fast calmodulin works is when you are looking at me and listening to what I'm saying right now, calmodulin is actually responsible for encoding the sights you're seeing and the sounds you're hearing into a signal that your brain can understand. So that means calmodulin has to work fast enough that it can encode all the changes in sound, all the changes in sight. I mean, think about how quickly you can see something move. Calmodulin moves even faster than that. So to understand what it's doing, we need methods that let us really freeze it and look at what it's doing really quickly. Okay. And so I mentioned this is that calcium, the closed structure without calcium. And when it has calcium, right, you can see the structure opens up and it changes. And those green globes there are the calcium ions attached to calmodulin. But it's not that simple, right? You think, oh, well, that looks easy enough to understand. Calcium comes in, changes its shape, so what? But that's not the reality. The reality is this is what the molecule actually looks like. Proteins and all molecules are constantly wiggling around. They've got enough energy, they're constantly fluctuating and jiggling and doing this and that. And it turns out that not just the very large changes in protein structure, but also all these little fluctuations are very important to how the protein works. And that's why we need these fancy physical chemistry methods to figure out what they're doing, because otherwise we don't really know what the protein is doing. The protein doesn't just exist as a freeze frame in one structure or the other. It exists as kind of this jiggly, flexible thing. And so now to give you a little bit more of an idea about what calmodulin looks like, I have prepared, I just have to share a different screen for you guys, so if you'll bear with me while I get this up. I made a little simulation video about calmodulin structure. So this blob, right, that's the structure you might have already seen of calmodulin before. That's the open structure. So let's zoom in. and explore calmodulin structure a little bit more carefully. So I'm going to dissolve that thing, and now we can see the molecular skeleton that makes up calmodulin. There's all these molecules knitted together into one complicated protein. And you can see all of the calcium ions bound in there. And now let's zoom in on just one of them, all right? We all right with that? Let's zoom in. So I'm going to zoom all the way in to one of these binding sites that calmodulin uses to sense the protein. We zoom in. And so this is a pocket that calmodulin has designed to actually bind calcium. And calmodulin is really good at binding calcium. It's better than almost anything else in your body at binding calcium. And you can see there are all of these atoms that participate in binding calcium. The protein is designed so that it's really strong at binding calcium. And all the red molecules are oxygen atoms. And so those are all there for binding calcium. Now we can look at another binding site. And you can see something that's really cool is this binding site looks almost exactly the same as the other one. And it turns out that these calcium binding sites in calmodulin in your body are super reproducible. All of us have 3 different genes that code for calmodulin. And if you make a tiny error in any one of them, it'll probably kill you, to be honest. I mean, the fact that we're all sitting here and living here means we'll probably have good calmodulin. But you have to be really careful about, making sure that calmodulin structure is right. Now, I mentioned calmodulin works with other ion channels. So I'm switching to another view of calmodulin, and I'm going to show you, you can take 4 calmodulins. You can take 4 calmodulins and you can stitch them together with 4 other proteins. And that makes one of these transporter channels that exist in our cell membrane. And if I change the view, you can see the channel that the protein goes through. I think it's going to, it'll change again. So we can see, you guys see that hole right through the middle? Oh no, it stopped at the worst possible time. Let me see if I can, let me go back to the end there and see if I can freeze frame. Yeah, so if you look at that, this is a big, huge protein that calmodulin is a part of, and you can see the hole. And based on if calmodulin sensing calcium or not, it closes or opens the hole. So that's kind of a little tour of calmodulin. We can now return to our normally scheduled programming. All right, so that's the molecule we're studying. Now we can ask, how do we study it, right? And so I already told you about this spectrometer. And so we built this down in Texas from scratch to study proteins like this. So we can do it in liquid, and we can do it with the really high time resolution that we need to get at this molecular information. So how do we do it, though? You can ask, how do we do it? We carry out a technique called infrared spectroscopy. Has anyone heard the term infrared before? And so I'm getting a mix of yeses and nos. And so infrared is basically, it just means a color of light that's even redder than red, right? And so infrared spectroscopy, or infrared light, is really good because it's very sensitive to the structure of molecules. And so what we can do is we hold our calmodulin or any other sample in a sample holder, and we shine infrared light through it. And based on what colors that molecule absorbs or doesn't absorb, we can learn a lot about its structure. And so what we do is we add up all the information we know about how it absorbs or doesn't absorb light, and we stack up that in a line, and that's called a spectrum. We have frequency, or color, I should say, along one axis, and how much light gets through along the other axis. And it looks like just kind of a confusing shape, but it contains a lot of information about the spectrum, of course, it is the spectrum, contains a lot of information about the structure of the molecule. And while that molecule is jiggling around really fast, that shape will change a little bit, and that tells us, is the protein wiggling or is it waggling? Did it zig or did it zag? Is it turning left or turning right? There's a lot of information we can get about the protein structure that way. All right, but I was talking about, well, we need that protein to slow down so we can watch it. And I just want to ask the chat, do you guys think of any good ways we can slow the protein down? Freeze it. That's a good one. And in my current work, that's actually what we do. We cool molecules down really cold, so they slow down and we can catch them in the act. But that's not actually what we're doing here, because remember, it's on the liquid phase. Add collagen, don't know at what temperature. I'm not seeing what I want. So let's imagine this. You're watching football and you want to see like right when the guy caught the football. How do you slow that football player down so that you can, see what he did there? Yes, someone said a picture. And that's absolutely right. We take a picture of calmodulin, but we don't take a normal picture. We use a laser pulse to take an infrared picture, and we get a spectrum that I just showed you that corresponds to a slice in time. You might ask, well, what kind of slice in time? How slicey are we talking? How thin is the slice? And to kind of illustrate this, this is a series of photographs that was taken in 1878 by someone named Edward Muybridge. It turns out before this guy took these photographs, no one knew how a horse ran. And so he had a camera and he took a series of photographs and he slowed that horse down. And so doing infrared spectroscopy is very similar, but we're not doing normal photography. This is what's called ultrafast. And if you were around last summer, you may have heard someone earlier give a talk about ultrafast spectroscopy. So how fast is ultrafast, right? It's going to blow your mind. So a normal photo, the fastest you might be able to get is a millisecond, right? A millisecond is about 1,000th of a second, okay? Ultrafast spectroscopy works on femtoseconds. So what's a femtosecond? Very few of us probably heard that word, right? So a millisecond is 1,000th of a second. A microsecond is 1,000th of a millisecond. A nanosecond is 1,000th of a microsecond. A picosecond is 1,000th of a nanosecond. Finally, a femtosecond is 1,000th of a picosecond. So if you ask, how thin is the time slice? It's pretty darn thin, right? We're dealing with femtoseconds, so you can see almost everything that happens while that protein is working. All right, so now kind of moving into the home stretch. So finally, you accept, we have our spectrum. We can collect a spectrum and the time slice on. We can slow this protein way down, ultra, super, slow-mo. But how do we turn that into a structure? Because that curvy line is nice, but it doesn't look like a protein to me. And so the answer is we do this with the help of simulations. So we can take an experimental spectrum with our spectrometer. We can also calculate an expected spectrum based on what's called a molecular dynamics simulation. And earlier I was showing you that protein wiggling around, right? A molecular dynamics simulation captures that motion and simulates, well, how should the protein wiggle and waggle? So we can take that simulation and we can calculate an expected IR spectrum. So then what we can do is we can compare that expected spectrum to our experiment. And so in blue, we can say is the expected spectrum and red is the experiment. And so sometimes the experiment and the simulation match up really well. And when that happens, we can say, awesome, looks like the simulation got it. Now we can say stuff about what that protein is doing. But more often, I'm sorry to say, the experiment and the simulation don't match terribly well. And in that case, what we do is we compare them, and then we change something about the simulation. Maybe we change how the protein moves around, or we change the temperature a little bit, but we mess with the simulation somehow. And then we run the simulation again. And this forms a loop of simulate, calculate, compare to experiment, and then refine the simulation. And this is how we can learn how proteins are actually moving around on this super duper slow-mo time scale. We can learn all kinds of important things about how you hear me talk, right? How does your heart know how to beat? Some people who have problems with their nerves or with their brains, what's going on there? Some people have problems with their hearts. What is the molecular reason at the smallest level that's happening? And so this is really the ground floor of figuring that all out. And so I kind of basically just said, and you guys saw the video. So using these methods, we can see what calmodulin is doing over, as calcium binds to it. And then you guys saw in that video, we can explore how on the right hand, that green calmodulin opens itself up and then hugs on to another protein so it can tell it what to do. And so, yeah, I think that's pretty much all I have to say. I saw a really good question in the chat. Someone said, how good of a computer do you need to do a simulation? And it varies. For something complicated like calmodulin with other proteins, you need a supercomputer. But, for simple molecules, if you just wanted to play around, you could do molecular dynamics simulations on a laptop. And the other great thing about a lot of molecular dynamics software is it's free. So you could download this stuff that professors and postdocs like me and graduate students put together and try it out for yourself. All right. So with that, I want to say thanks very much for your attention. I love all the activity in the chat. And I also want to say thanks to the groups that I've been very fortunate to work with. The BIAS group down at UT Austin, you can see in that picture I'm wearing, this might be the same shirt. I like this shirt a lot. And then also our Zoomified Johnson group. So with that, I'm sorry I went a little bit long, but thank you very much for your attention. I don't know if we do questions, but I'm happy to take any. And that's all I had to say.
Yes, we do questions. Thank you so much, Sean. This was a great talk. You can see in the chat all the thank yous you're getting from the crowd. And so while you were speaking, you did a pretty good job answering most of the questions, but we took down all the rest that you didn't get to. And if you have any other questions for Sean, make sure to put them in the chat right now and we'll see if we can get to them right now. So one of the questions that we had in the chat was, does calmodulin only respond to calcium in this way, or are there other proteins that work the same way?
Oh, yes, that's a great question. But hold on, I just have to adjust my audio for a second. So there's kind of two questions there. Is it okay if I stop the share?
Oh, absolutely. You can stop the share.
Or maybe I already did. Actually, now I'm looking at it. Yeah, so anyway, the cool thing about calmodulin is it doesn't just respond to calcium, it can also respond to pH, which is how acidic your cell is. So it also cares about pH. But I think the bigger part of that question is calmodulin the only molecule that responds to calcium? And the answer is no. Calcium is used as a signaling molecule in a lot of processes. In fact, your cell spends a lot of energy getting rid of all of its calcium. If you look inside the cell, the concentration of calcium is like 1,000 times lower than many other ions that you have in your blood. And that's because the cell wants to be able to use calcium as a really good communicator, right? If you're listening to someone's voice, you want it to be really quiet so you can hear their voice really well. And it's the same with calcium. But anyway, calmodulin is not the only molecule that responds to calcium. There are many molecules that respond to calcium, but calmodulin may be the most important. And it turns out that calmodulin controls over 300 other proteins in your body. So that one protein calmodulin is talking to literally hundreds of other proteins. So that's why it's such an interesting research target. It's really in the middle of a lot of what's going on in your cell.
Nice. Thanks so much for that answer. Also, while you were showing the calmodulin simulation, there was a lot of discussion in the chat about how the protein looked. Some were saying it looked like gum. Some were saying it looked like dogs, like pills. Can you talk a little bit about the design of like how the molecules look like? Do they really look like dogs and pills, or is that more of an illustration?
Yes, I mean, it's a bit of a tough question because the molecules are so small that there's no real analog for what they really look like. Because the way a molecule, right, when you're looking at me, light bounces off me in a certain way because I'm pretty big. But calmodulin is tiny. And so you can't really say that it's really hard to say how it looks because light interacts with tiny molecules in a way that's totally foreign to us. But what I can say is those pictures based, of what calmodulin looks like, a lot of them are based on a kind of photography called X-ray crystallography. And so when you look at calmodulin, it doesn't really have color in the usual sense, but it does have structure. And so those structures I was showing you, right, if you could look at an outline of calmodulin or kind of a 3D model of calmodulin, you couldn't say much about the color in kind of human terms, but you could say that structure is quite accurate. And the way that structure comes to be is the process of evolution, right? You guys know that as we evolve, just kind of by chance, right? Some random change happens and it turns out that that's useful to the organism. So that organism does really well and it makes many copies of itself. And that's the same way calmodulin evolves. And so the way calmodulin was set up, I was talking about, there are those 4 binding sites that you saw in the video. And those binding sites are highly evolved. Those are really optimized by evolution to bind calcium in a certain way. And so calmodulin's appearance or its structure, it's all about, number 1, making those binding sites as good as they can be. And number 2, you guys saw there's that central area of it that it uses to hug onto other proteins. It's also about making that central area the right shape so that it can hug on other proteins and give them instructions about what to do.
So we just actually got a question related to that. If you didn't have like all the calmodulin that you needed to form that shape, could you survive? Like if it was somehow missing one?
You mean if it was missing one of its ion binding pockets, or if that ion channel didn't have the calmodulin?
I think what they're asking is if it was missing, like when you were building the protein and you showed the ion channel, I think they were asking what happens if you were missing one of those pieces? Could you still survive?
Yeah, so the answer is almost certainly not. Now I don't know too much about that multimeric. And when I say multimeric, I mean it's a structure that's formed out of multiple proteins. I'm not a biologist, so I can't say for certain what's going on. The structure is a nice illustration. But just like calmodulin evolved, that multimeric protein evolved very specifically to incorporate all 4 calmodulins. And so now if you had just one of those proteins that was missing one of them, you'd be fine because you've got trillions of cells. But if somehow all of your proteins are missing one calmodulin, that ion channel couldn't work, and your cell wouldn't be able to regulate, what's coming into and what's coming out of it. And so that would be a big problem. A related question is, well, calmodulin has those 4 binding sites, and what happened, if one of those gets messed up? And you saw, the calmodulin is a pretty big molecule. It's got a lot of structure, but those binding sites are super important. And it turns out if just one of them is mutated just a little bit, so that one of those oxygen atoms is not quite in the right place, it can be really serious. And so there's a whole class of diseases called cardiac arrhythmias, where somebody's heart doesn't beat quite right. And a lot of these are caused by calmodulin, and they are very frequently fatal. So calmodulin is a great example of how, molecules in our body can be tuned really, really carefully. You can't mess with it, or everything, everything stops working. But usually a lot of these problems with calmodulin, they're called calmodulinopathies, right? They often, they happen very early on in development. So if you have got a real problem with calmodulin, it's probably going to take you out, very early in your life. You wouldn't have made it this far. So no one needs to go around worrying if their calmodulin is all right. Just the fact that you're sitting here and talking with us right now means that calmodulin is doing okay.
And you mentioned that it can affect your heart, for example. And one of the questions we had in the chat is, where is the calmodulin located in our body?
Yes, that's a great question. And so the answer is calmodulin is located all over the place. In your cells, for example, I would say, a lot of proteins that are doing your jobs are located in your cells. And I just want to remind everybody, I am a physical chemist. I'm on the physics side of chemistry. So I'm not a super expert in the fields of biology. And if anyone is and I misspeak, I hope I will be interrupted and corrected. I'm a little bit out of my expertise here. I'm all about building lasers. You guys saw the lasers I built. It takes a long time. But to answer the question, so calmodulin's all over your body. It's in every cell. And so, but where actually is it located? So for example, in your heart's made out of muscle, right? And your muscle's made out of muscle cells. And so calmodulin is located inside the muscle cells. And so the calmodulin will sense calcium and tell the heart how to beat. Calmodulin's also in your nerves. You may have heard the term neuron or axon or dendrite or nerve. And these are all part of machinery. Your body is essentially full of little wires that transmit signals, not with electricity, but with chemicals. And so calmodulin is integrated into all of those nerves and all of your synapses. It's all, in your brain and everything. And it's, keeping track of those signals and helping to generate those signals. So I'm sorry, it's not a very satisfying or specific answer, but it's all over the place. And the cool thing about calmodulin, it's not just in you. If you've got a dog, your dog's full of calmodulin. If you've got a cat, I saw in the quiz, those cats are full of calmodulin. Cows and roosters and all the barnyard animals have tons of calmodulin. And you can look up this word, eukaryote is a term for a certain kind of cell. And so we're made up of eukaryotes, all mammals are made up of eukaryotes, et cetera, et cetera, et cetera. All eukaryotic cells depend on calmodulin. So it's really like it's everywhere. You can't turn around with, you can't avoid it. You can't avoid calmodulin.
And yeah, sorry to inundate you with all the biology questions, but we do
I'm
have
happy to answer as well as I can.
we do have more of a physics question. What defines a supercomputer? What makes it so super?
Yeah, that's a great question. That's a really great question. And so supercomputer it's a very ambiguous, it's a vague term, and because the term supercomputer changes with time, right? If you have a phone, this is an iPhone 5. This is an old iPhone is what I'm trying to say, because I don't like the new technology. But this old iPhone, if you took this, I don't know, 20 years ago, this would be a supercomputer. So you always have to be careful about the term supercomputer, because there's this thing called Moore's law, which means that computing power, the density of transistors, can increase. It's on a doubling, it's on an exponential rate. But when I say supercomputer, what do I mean? I mean to run a simulation of a complicated molecule like calmodulin, right? Imagine a powerful desktop computer. Something you might, I don't know, play a new, game on, something that has really good graphics. It turns out that today, back in the day, back in the 70s and the 80s, a supercomputer was one huge computer that a bunch of engineers put together with bolts and wrenches and so on and so forth. Today, supercomputers usually aren't built that way. Supercomputers are usually built as clusters or networks of many smaller computers. And so a supercomputer that I might run simulations on is a network of, something like 1,000 really powerful desktop computers. And of course, it's not exactly a desktop computer. There are, some differences in the hardware, but it's essentially, it's like a desktop computer. And so a university might have a couple huge supercomputers with, 1,000 of individual network computers. Each of those network computers, when it's in a supercomputer, it's called a node. And when I run a simulation of calmodulin, I don't use the whole supercomputer at once. I'd have to wait a long time to do that, or they might never let me do it. I might use 10 nodes at once. So whatever, 10 powerful desktop computers, if you could reasonably call that a supercomputer, or just like a sub supercomputer. Like, we're not talking about Superman, we're talking about like Batman kind of supercomputer. In terms of raw power, I mean, Batman is the world's greatest detective, of course, but he doesn't have laser beam eyes. So I hope that was a helpful answer.
Yes, and so we just have one last question for you. Back to your work specifically, some students were wondering, how long does it take to build those lasers and those cool machines that you'd made?
Yeah, so that's a great question, and it varies a little bit because it depends on what kind of machine you're trying to build and what kind of resources you have. So the ultra-fast spectrometer I showed you down in Texas, it's on the laser table and everything with the black sides. That's a more advanced kind of instrument, and I didn't build it myself, right? There's my boss and assistant professor helped me build it, and there's a very talented graduate student who is now herself a postdoc who helped me build it. And so I'd say that took all of us, I don't know, like 8 months of full-time work to actually get it working. And I mean, the cost is also important. That it was something like, I would say $600,000 to build that device. And so that's, as far as scientific equipment goes, I'd say that's in the mid-range. Because it's not like we were building like a particle accelerator. We weren't building a telescope to shoot up into space. I mean, that's even crazier. But some of the other stuff, I built by myself, and I didn't take too long. So the thermal desorption spectrometer that I built in graduate school, that's not as complicated a device. And so I was able to build that out of spare parts in our lab. And I don't know, like, I wasn't working on it full time. If I'd been working on it full time, it would have been, I don't know, like a few weeks to a month. So it really depends on what you're building. But the thing I want to point out to everybody is you can build a useful and interesting scientific device. If you just want to investigate air pollution or temperature and carbon dioxide concentrations. If you had $100, I could give you a list of stuff you could build, and if you took a couple weekends, you could make a pretty cool device that would actually be useful in measuring things like air pollution. So there's a real range, and it has to do with resources and what you're trying to do and how much time you have to do it.
Well, thank you so much for talking about, the time that you dedicated to your instruments. And also thank you so much for taking your time tonight to talk to all of us. We really enjoyed it. And we are going to send it to another volunteer to close out the meeting today. But thanks again, Sean.
Yeah, thanks. And the last thing I want to say is just, I want everyone, science sometimes can seem scary. It seems like you have to be a super genius to do it. And I want to tell you, in my experience, it's never about how smart you are. And that means different things to different people. It's about how persistent you are and how much, how hard you try to stick with it. And so I strongly believe, if you stick with it, anyone can be a scientist. So just remember that, no matter how discouraging some other people might be, it's all about effort and persistence. So it's a real pleasure.